Good afternoon, dear participants. Welcome you all in the afternoon session of today's session. As I can see, our next speaker has already joined. Sir, am I audible?
Yes.
Yes. Sir, may I proceed? Okay.
Yes.
So it is my pleasure to introduce Professor Subhash Bhattacharya, an eminent chemist and distinguished adjunct professor, Department of Applied Science, Macau, West Bengal. He is also the former professor of Department of Chemistry at Jadavpur University. Over several decades, Professor Bhattacharya has made significant contribution to physical chemistry, particularly in the areas of photochemistry, surface chemistry, nanomaterials, and the photophysical behavior of photoactive molecules in bionano cavities. His works explore how molecules behave in confined nanoenvironment,
হ্যাঁ করেছি
an
তো
area with wide
করেছি
implication for material science, photophysics, and emerging bio-inspired technologies. At Jadavpur University, he has served with distinction as Dean of Faculty of Science and later as the head of the Department of Chemistry, helping to shape both academic leadership and advanced research culture in the department. Professor Bhattacharya has also carried out Jew's reputation abroad through visiting position at foreign institutions, fostering international collaboration and scientific exchange. His teaching, mentoring, and research have influenced generation of chemists, and his contribution continue inspire new direction at the intersection of chemistry, material science, and nanotechnology. We are honored to have him with us today. Welcome, sir. This platform is now yours.
Thank you for your introduction.
Welcome, sir.
Good afternoon to everybody. Now I proceed directly to my lecture. I shall discuss about soft chemical techniques for the synthesis of nanoparticles. Okay. Is it visible?
Yes, sir.
Okay,
Yes, sir.
thank you.
Yes, sir.
So my topic,
Sir, excuse me, sir. Make it full screen mode for better visibility. Slideshow.
slideshow. Uh,
Sir, press F5. F5.
F5. Okay. Slideshow.
Sir, in the toolbar you can see slide, top.
Slide, yes,
Click on it.
I am doing, but what happened actually? Slideshow, share. Okay. Shall I do it again? Just a minute.
Okay, sir.
Google Drive. Now?
Sir, it's not visible, sir.
Not visible?
Yes.
What happened? Share. Google Drive. This. Share. What happens? Now it is in?
Your desktop is visible. Now you have to open your PPT.
Now?
No. Sir, it is in the right top. It is there is a tab slideshow.
Slideshow, yes.
Yes. Start. Click it. Yes. Now it's okay.
Now it is okay?
Yes, sir, it's okay.
Thank you. Technical difficulties. I face problem.
No issues.
Sorry for your wait. Okay. It is approach towards understanding soft chemical synthesis of nanomaterials for diverse applications. Actually, uh, nanomaterials, uh, its synthesis and its functionalization is a technique. Uh, it will be helpful to one depending on your uh, choice of functionalization. Now, we generally nanomaterials are synthesized by two processes. One is top down, another is bottom up. Generally, physicists use top down, chemistry persons, and now most of the physicists also use bottom up approach. So, from chemistry viewpoint, uh, we have used bottom up approach, that is understanding soft chemical synthesis of nanomaterials for diverse applications. That means the nanomaterials which were synthesized that can be used for different purposes, different applications. I shall discuss one by one. Why soft chemical approach? First point, it is easy process among different processes, easy ways to introduce ligands. That means functionalization. For a bare nanomaterial will be of no use if it cannot be functionalized. That means proper ligands are not introduced. To introduce ligands on the surface of products, they are good for their protection from environment so that they do not aggregate. That is, if a bare nanomaterial is covered by different ligands attached on the surface, then another nanomaterial will not come nearby. So they will not aggregate by dispersing them in either in aqueous or organic media. Synthesis factor such as raw material, acidity, solvent concentration, additive, temperature, and reaction time. These also can be changed, can be manipulated according to our convenience to direct the shape and size of the final products. That means it is advantage for your synthesis of desired nanomaterial. Third point, these routes show multiple advantages. What are these? Low temperature, simple apparatus, low cost, potential for the large scale production, and versatile post treatment. That means, first point, it is easy to attach ligands on the surface of the nanomaterial for functionalization according to our choice. Second point, cost effective, because raw material is easily available and of low cost. Acidity, solvent concentration, additive, temperature, reaction time, that can be changed according to our wish to shape the size, to direct the shape and size of the final products. And last point, temperature must be low, simple apparatus, just test tube, beaker, etc. and large-scale production also occur, and versatile post-treatment. Now, what are the different soft chemical approaches? There are different soft chemical approaches. One is hydrothermal method, second soluble method, third ionic liquid method, ionic liquid based method, dendrimer using method, gamma radiolytic, and biopolymer based. These are the soft processes. These are the soft processes. Uh, and during these processes, the media which we accept for the soft chemical synthesis, that is there micelle, reverse micelle, vesicle, cationics, liposome, block copolymer, biosurfactant at air-water interface. We have tried all the methods, we have tried all the media for the synthesis. But now I here discuss only reverse micelle method, cation ionic method, and another is gamma radiolytic method. These three methods I shall discuss here. So using this surfactant, using this micelle, reverse micelle for this production, for the preparation of micelle, reverse micelles, vesicle, cationics, surfactants are required. These surfactants may be cationic, anionic, or non-ionic. These are the requirements which will be needed for synthesis of nanomaterials by these processes. Acha. Now, according to applications and wide applications, we have chosen phosphate nanomaterial, generally lanthanum phosphate nanomaterial we have synthesized because its applications is wide. So first soft chemical reverse micellar method has been adopted for synthesis of nanomaterial. Reverse micellar method is under soft chemical process. It has been taken for the synthesis of nanomaterial. Reverse micelle, what now definition of reverse micelles? Chemistry participants must know, but other participants are also there, so I shall discuss now. What is reverse micelle? A surfactant may be anionic or cationic or non-ionic. When it is dissolved in non-polar organic solvent, that means non-aqueous, non-polar solvent, then reverse micelles are formed. Micelles are formed when surfactants are dissolved in water. And when surfactants are dissolved in organic solvent, non-polar organic solvent, then micelles are reversed, that is reverse micelles are formed. In micelles, we know core is hydrophobic and outside is hydrophilic. But in case of reverse micelle, core is hydrophilic and outside is hydrophobic. You see in this figure, that is a surfactant has a long chain tail of hydrocarbons. These are, and a hydrophilic hydrophilic head. In normal micelles, heads are outside, tails are inside. Whereas in reverse micelles, tails are outside, heads are inside. Now, to make the process easier, to make the formation of reverse micelles easier, drops of water are added there. Drops of water are added there. Why? Drops of water go straight to the inner side because these heads are hydrophilic. Water is hydrophilic. Sorry, water will be loved by this head group. They enter here. Water molecule come through this channel, enter here. And the core is here. Within the core, it is hydrophilic. Water is there. So, during the formation of reverse micelles, you require surfactant, cationic, anionic, or non-ionic, and organic solvent. Generally, we take heftane or hexane. Then, during mixing, add some drop of water. The water will go here and it will adhere the head groups so that easily reverse micelles are formed. But one point, sometimes when surfactants are dissolved in non-aqueous solvent, they do not easily dissolve. Some co-surfactants are added. Some co-surfactants are added, generally isopropanol or ethanol, but very little amount. It must be less than 5% or maximum 10%, but above 10% it will be destroyed. So co-surfactant sometimes needed to dissolve the surfactant in non-aqueous solvent. Am I clear?
Yes, sir.
Yes, 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 sir.
Okay. So, in our case, no co-surfactant was added because the surfactant was easily soluble. And the amount of water present here, amount of water added here, that is called a water pool. It is represented by omega. Omega equal to concentration of water by concentration of surfactant. Water pool, actually number of moles of water by number of moles of surfactant. That means concentration of water, concentration of surfactant. In reverse micellar case, generally water pool omega value lies between 3 and 4. If more water is added, if little bit more water is added, then these are not reverse micelles, then it becomes microemulsion. Then this becomes microemulsion. And if more water added, then it breaks and emulsion is formed. For example, Dettol, when it added, emulsion is formed, and that is due to this reason. Now, actually, this water core, this water pool, it is the site of reaction taking place here. Reaction occurred here. So, some surfactants, for example, titanic sunrate, it takes little amount of water. So, not suitable for a reaction to prepare more product. That is why you have to choose such a surfactant which can take up maximum water. We have chosen an anionic surfactant, aerosol orange tea, AOT. AOT, organic solvent, is heftane. When AOT is added in heftane, no co-surfactant is needed. Dissolve AOT in heftane, add water, and it will form reverse micelle. It cannot be seen by visible eye. That is why it is called pseudo phase, two-phase system. Another phase that cannot be visible called pseudo phase. So reverse micelles of AOT in heftane is formed by adding water, and AOT can take up maximum amount of water. Omega is high in the reverse micellar region, and if more water is added, then become microemulsion. So the water core will be large, more amount of reactants will be entered here, and reaction takes place. And look and think, if we add, so reverse micelles are formed, AOT, aerosol orange tea, heftane, and water. You take a beaker. In the beaker, add heftane, add AOT, surfactant, shake, and then add drop by drop water. Reverse micelles will be formed, and its shape will be like this. These are chemically inert outside the reverse micelles. There are so many reverse micelles will be produced there. They have Brownian motion, unique microenvironments, controlled size and shape, chemically inert. And interesting, this core range is nanometer range. That means the dimensions of the water pool is nanometer regime, 1 nanometer to 100 nanometer within this. So if you synthesized products here, their size will also be nanometer sized, because they are forming here. So their size will be within the nanometer range. This is the most advantage. Hope it is clear to all of you. Now, next. I have told you that lanthanum phosphate we will prepare. Surfactant use AOT. This is the structure of AOT. Sodium 1,4-bis 2 ethyl hexoxy 1,4-dioxobutane 2 sulfonate. Trade name is AOT. Aerosol orange tea. So we have prepared reverse micelles. We add more water. That is why it becomes microemulsion. Microemulsion. What we did, instead of water, we have not added water. We have taken N-heftane in a beaker. We have actually taken 2 beakers. In one beaker N-heftane, in another beaker also N-heftane. In one beaker AOT added, in another beaker AOT added. Now we have to add water to form reverse micelles. What we did? We did lanthanum nitrate solution and disodium hydrogen phosphate solution into different test tubes. In 2 test tubes, in one test tube we have prepared lanthanum nitrate solution, aqueous solution, and in another test tube disodium hydrogen phosphate aqueous solutions we have prepared. Now in the reverse micelles, instead of water, we added lanthanum nitrate solution in one beaker and disodium hydrogen phosphate solution in another beaker. That is, microemulsions are formed in 2 beakers, and within the core, one is lanthanum nitrate, and in another there is disodium hydrogen phosphate. That is microemulsion 1, microemulsion 2. Now, Room temperature. No, there is no question of temperature, room temperature. Now, what we did, microemulsion 1 drop by drop added to microemulsion 2. That is, one solution in a beaker is added to another solutions in another beaker. Now shake with glass rod, you shake. Mixing of microemulsion 1 and 2. During mixing, what happens? This droplet collide. These droplets collide. Droplets of one beaker adding into another beaker collides with droplets of another beaker. And that it is like fusion reaction. That is fusion reactions. So lanthanum nitrate and sodium hydrogen phosphate come close to each other and reaction occurs and produce lanthanum phosphate. So our procedure is simple. Take 2 beakers. In 2 beakers take heptane. In 2 beakers add aerosol orange tea, shake, and then add in 1 beaker add reactant aqueous solution. That is 1 reactant aqueous solution, lanthanum nitrate aqueous solution. Another beaker add disodium hydrogen phosphate aqueous solutions. Instead of water, aqueous solution of that. And these two go straight into the core of the reverse micelles, that is in the water pool. Now add solutions of 1 beaker to another beaker. Now the collision takes place, fusion reaction takes place, and lanthanum nitrate and disodium hydrogen phosphate reacts and produce lanthanum phosphate. Chemical reaction occurs, and these black spots are lanthanum phosphates droplet, and these are within the core. That is, their size must be within the nanometer range. And if we shake vigorously, the droplets will break and the lanthanum phosphates will precipitate. After the collision, they mix and produce the desired product, and they precipitate it. Okay, next. That is, precursor chemicals, lanthanum nitrate or cerium nitrate. Oh, here, there is another point. So lanthanum phosphate nanomaterial will be produced. But here also, what we did, cerium and terbium, cerium ion and terbium ion, we also added into the nanomaterials so that the cerium and terbium entered into the cage of lanthanum phosphate. That is, aqueous solution of lanthanum nitrate, some cerium nitrate and terbium nitrate. The 3 mixtures are added. And phosphate, any one of them, disodium hydrogen phosphate, sodium hydrogen phosphate, etc., any one, phosphoric acid, any one. Oil medium, organic medium, N-hexane, N-heptane, cyclohexane, N-octane, isooctane, N-decane, any one of them. We have chosen N-heptane. We have chosen disodium hydrogen phosphate and lanthanum nitrate with traces of cerium nitrate and terbium nitrate. Surfactant used AOT. Double-tailed anionic surfactant means double-tailed means that is the with head group 2 hydrocarbon chains. Now look here, AOT plus organic solvents, any one of them, plus water. Instead of water, we have added the solutions of reverse micelle 1, lanthanum nitrate, terbium nitrate, cerium nitrate, and here any one of them. And omega, water pool is kept within 5 to 30. We generally we have taken omega equal to 25. And this work have been published in Journal of Physical Chemistry, C. Now, it is mixed, shaken, starred, and that is within the core. Lanthanum, cerium, terbium, phosphate comes and white precipitations. 4 hours stirring, room temperature, white precipitation, centrifuged, washed, dried in vacuum. The precipitates of lanthanum phosphates within its lab, cerium nitrate and terbium nitrate taken. The products are formed, white precipitate. Now, after shaking, it is centrifuged so that the precipitate can be separated and dried in vacuum. Sometimes one of my students initially for quick process he entered into the oven. That is all destroyed. That again done. So lastly precipitate must be dried in vacuum. And the products are formed. It is nano rods, nano-shaped rod. Why phosphate nanomaterials? There are so nanomaterials. So why we have chosen lanthanum phosphate nanomaterials? These are the reasons. It can be used instead of semiconductor quantum dots. It is fluorescent alternative. Fluorescent. These are fluorescent. Where during applications you can use its fluorescence properties. Used as cellular markers, demonstrating localization in the cytoplasm of cells with no induced toxicity after internalization. That is, within the cell, human cell or any animal cell, to culture a tissue, to diagnose the tissue, you add some lanthanum phosphate there by some tape or by some injection, and they will they are entering into the tissue, they fluoresce, and showing its fluorescence property, you can diagnose, you can detect is there any disorder in the system, and they have no toxicity. Provide a rigid crystal environment for the dopant ions. That dopant ions, that is cerium and terbium and dopant, they are protected there, rigid crystal environment, resulting in a higher photoluminescence quantum yield. They are also effective laser materials, can be used as catalysts, heat-resistant materials, proton conductors, versatile biological levels in living cell, and photon up conversion materials. These are used for these purposes. And here, lanthanum phosphate with cerium and terbium in its core, highly efficient and commercially applied green phosphor in fluorescent lamps. This is used in fluorescent lamps as green phosphor. In signaling system, there is green signal is used, they are used. Next. The phosphate materials are prepared, lanthanum phosphates. So we have to characterize, otherwise without characterizations, you cannot claim anything. Because you have to, students and research scholars and scientists will first try to know what is the characterizations, whether it is really lanthanum phosphate. That is, we have done XRD and IR also, FTIR. From XRD, different diffraction peaks observed, and from the different observed peaks, it is monoclinic type. Their structure is lanthanum phosphate with cerium terbium within its core as dopant ions. They have rod-like structure, nanorod, and monoclinic structure. So monoclinic, that is one axis 8.25 angstrom, another is 7.09, another is 6.42 angstrom. And এই কার্ডটা ইউজ করে, these cards have been used Using this, we have found this. Now, without cerium terbium, the lanthanum phosphate nanorod, and with cerium terbium, lanthanum phosphate nanorods, there is no significant change. That is, lanthanum phosphate, when used as host of cerium and terbium ion, there is no change in structure. But slight shift in the peaks are observed. That is due to cerium ion and terbium ion, because their size almost identical, 1.14, 1.14, 1.09, 1.15. There is the, it depends on the ratio of the size. So peak size may differ slight. Next, TEM, transmission electron microscope structure, picture of the nanorod which I have prepared. This is the nanorod. Stem structure, stem picture of the lanthanum phosphate nanorod. This is said pattern, which layer from which layer comes, and these are the actually nanorods. After TEM analysis, we have found when these solvents are different, N-hexane, N-heptane, N-octane, N-decane, cyclohexane, isooctane, and lanthanum phosphate, cerium terbium within it, all forms nanorods. Their length, width, this length and width. And the ratio of length and width is called aspect ratio. For a nanomaterial, the ratio of length and width is called aspect ratio. Depending on their aspect ratio, activity differs. Depending on aspect ratio, activity differs. So activity of nanomaterial depends on aspect ratio. So here AOT concentration 0.02 moles per liter, and when 0.5 moles per liter, the all in both cases nanorods are formed, but their size are different. Aspect ratio also different. And maximum when omega 10, instead of nanorod, nanoflower is formed. We have seen in the this is nanoflower, omega 10 nanoflower. Okay, so TEM analysis using different solvents and different omega, we have prepared lanthanum phosphate with cerium terbium ion within it, and the sizes like this. Now, a spectroscopic study, lanthanum phosphate, lanthanide phosphate, lanthanide photophysics within it is a broad chapter, lanthanide photophysics. Actually, lanthanum phosphate, cerium ion and terbium ion have taken. Within lanthanum phosphate, there are cerium ion and terbium ion. What happens? First, consider cerium ion. Its ground state is 4F1 structure, and from spin orbit coupling, its nomenclature of the states is 2F5 by 2, 2F5 by 2, and 2F7 by 2. And when it is excited, when cerium excited, then 5D, 5D orbitals, and from spin orbit Russell-Saunders coupling or spin orbit coupling, the nomenclature is 2D 5 by 2, 2D, 2D 3 by 2. When lanthanum phosphate is with cerium ion and terbium ion is excited at the wavelength where cerium ion absorbs only, not terbium, where cerium ion absorbs only. After absorption of radiation, cerium ion excited goes to 5D orbitals from 4f1. And then it emits radiation, that means fluorescence occurs. And fluorescence when occurs, that is from 2D 5 by 2 to comes 2F 5 by 2, or 2D 3 by 2 comes to 2F 7 by 2. That is the fluorescence emission occurs at 342 to 341 nanometer and 460 to 450 nanometer. Most intense one at 340, and we use 340 nanometer. So what we did? In the lanthanum phosphate, there is cerium ion and terbium ion. Now we excite the nanoparticles with the radiations where cerium only absorbs, that is nearly 270 nanometer. Cerium ion only absorbs. After absorption of radiations, it's appropriate at absorption wavelength, the radiations absorbed and cerium ion excited to 5D orbitals, and from there it emits fluorescence and we get 2D to 5 by 2 strong emission, and then another also 2D to 2F 7 by 2. This is the curve, this one. At 340 nanometer, we have taken 340 nanometer, this one. This one. Now, consider terbium ion is also there. And what happens when cerium ion emits fluorescence, terbium ion accepts this. That is energy transfer. Cerium ion transfers its energy by fluorescence emission, and there is terbium ion. It accepts the radiations. And for terbium ion, ground state is 4f8. That is the nomenclature is 7f6. And excited state, it is 4f7 5d1 excitation level, that is D 5d4. To 7f, F has so many states, 0, 1, 2, 3, 4, 5, 6, these levels. So terbium ion absorption band 200 to 320 nanometer, and transition occurs from 4f8 to 4f7 5d1, 4f7 5d1 excited state, and then terbium, after taking radiations from cerium ion, it will be excited, and then after excitation it will emit. Now with increased terbium ion concentration, the emissions from 5d3 to 7fj levels were quenched gradually by the cross relaxation process between neighboring terbium ions. What happens? Terbium ion emission occurs, that is fluorescence emission occurs. Due to presence of maximum terbium ion, sometimes due to collisions with the nearby terbium ion, the fluorescence emission decreases, that is quenching occurs. It is called self-quenching or concentration quenching. Now look here, in case of terbium, 4-line emissions peaks obtained at 490, 544, 490, 544, 588 and 627. Among these, 544 is maximum. So you excited lanthanum phosphate where cerium ion take its absorption and it becomes excited and then emits fluorescence, and then that fluorescence emission taken by terbium ion, and terbium ion again emits. That means energy transfer occurs, cerium to terbium. Oh, achha. Oh, there is another point just before it. Uh, Up to that, terbium ion. Then just one point I forget to say. Achha. That is the fluorescence intensity. Fluorescence intensity. Uh, PL intensity means photoluminescence intensity. That is fluorescence intensity. It increases if the solvent decan is used instead of heptane or hexane. Now, the nanoparticles which are formed, their quantum yield differs. Quantum yield means uh, uh, fluorescence intensity divided by absorption intensity. It is called quantum yield. It varies from solvent to solvent. According to your choice of solvents, quantum yield differs. For decan, it is n decan, it is maximum. And CIE coordinates, that is in the theoretical literature, in the if you add the data in the CIE plot, then we will get like this. Depending on the CIE coordinates, that means one is composition, another is intensity. The value depends on this. If the CIE coordinates be 0.33, that means 0.3, 0.3, or 0.33, 0.33, then it becomes a white light. And depending on CIE coordinates, their light, their color differs. That is brown, yellow, cream, etc., green, depending on CIE coordinates. If the CIE coordinates of any product, depending on solvent, be 0.33, 0.33, then it will be white. We have seen the fluorescence intensity of the lanthanum phosphate containing terbium. This is. Next, energy. I have told you energy transfer between cerium ion and terbium ion. So cerium ion emits fluorescence and terbium ion accepts it. Within the cage, there are cerium ion, terbium ion. Cerium ion loses its fluorescence intensity, terbium ion accepts it, and so the reaction is energy transfer. It emits at 350 and terbium absorbs at 350 and emits at 545. And it is the time resolved spectra. Lifetime has been measured. These are used to find the mechanism of the energy transfer. Time resolved quenching of cerium ion luminescence with gradual addition of terbium ion. Next slide. Mechanism of energy transfer. That is, in case of cerium ion, it is excited and then energy transfers, and terbium accepts, and except from excited to come downstate. And the distance between them is 11.9 angstrom. Distance between them is 11.9 angstrom. Actually, this Blaise reaction, I have tried many times, but it cannot be written here. You will get Blaise equations in the literature, from where we have calculated the distance between them, and the in the equation used, V is the volume of unit cell, N is the number of host cations, X is the total concentration of cerium ion and terbium ions. So from the our results, the energy transfer is dipolar quadrupolar character. Generally, we know dipole-dipole transitions, dipole-dipole character, but here is dipolar quadrupolar character. Dipole quadruple transition occurs. So Foster mechanism will not be valid here, because Foster mechanism is valid for dipole-dipole interactions and for long-range interactions. But here, short-range interaction within the core. Here, Dexter mechanism has been adopted for short range interactions between dipole quadrupole interactions, and here n value becomes 8. So mechanism of energy transfer in lanthanum phosphate core having cerium ion and terbium ion is Dexter mechanism occurs with dipolar quadrupolar interaction. The mechanism of energy transfer, the distance between the reacting ions and the value of number of ions in the cell per unit n equal to 8 has been calculated. That is the mechanism of that. I have told you dipole quadrupole interaction 11.4 agreement with Blass equation. Okay. Now we have synthesized nickel oxide nanomaterials. Nickel oxide nanomaterials, in previous case for lanthanum phosphate we used reverse micellar microemulsion method, but here we have used cation ionics method. Cation ionics method. Here we have used cation ionics method. New method. I have told you cation ionic methods. 2 minutes. Hope up to this it is clear. Now it is quick, but I think it is clear to you. Okay. Am I audible?
Yes, sir. Yes, sir.
Okay, okay. So I think the up to that clear, eh?
Yes, sir. Yes, sir.
Yes, sir.
Now,
Yes.
nickel oxide nanomaterials I have synthesized using cation ionics. Cation ionics, from its name, I think you can understand that is two types of surfactants have been used. One is cationic and there is anionic. That is cationic surfactant CTAB, cetyl trimethyl ammonium bromide, known to you, familiar to you, and anionic surfactant sodium dodecyl sulfate. It is also common. So we provided an effective methodology for controlled room temperature aqueous synthesis of nickel oxalate, nanosits, and nanoflex in presence of anionic rich self-assembled bilayers of cation ionic surfactant comprising of anionic sodium dodecyl sulfate and cationic cetyl trimethyl ammonium bromide. When the two are mixed, they produce coservate, and that is called cationic. Using this cationic, we also prepare nanomaterials. Understanding the role of cation ionic surfactant in morphology controlled synthesis. What is our motive? What is our motive? Our motive was to synthesize nickel oxide nanomaterials. And using nickel oxide nanomaterials, we have used photodegradation of phenol. Why phenol? Because in most of our industries, the wastewater which comes out from the industry, they contain phenol because there are different organic reactions, and as a result, there are phenol produced. So in the wastewater, mostly there are phenol. So if we degrade the phenol, then it will be a valuable work. So our investigations on the photocatalytic activity of nickel oxide nanomaterials on the photodegradation of phenol under ambient condition explores an effective pathway towards wastewater treatment. To develop a simple, versatile, and environment-friendly method for the large-scale controlled synthesis of nickel oxide nanoparticles. And from here, we calculated the nickel oxides have also bacteria killing property. bacteria killing property. That is minimum inhibitory concentration. That is the minimum amount of nickel oxide required to destroy the um, um, microbes. That is called minimum inhibitory concentration. And bacteria killing minimum bactericidal concentration, MBC, over gram positive and gram negative bacteria. We have calculated. And to investigate the hemolytic activity for understanding influence of nickel oxide nanoparticles on human health. Why nickel oxide is one of the relatively few metal oxides with p-type semiconductivity. It has p-type semiconductivity. It is used as a catalyst, as electrode material for lithium-ion batteries and fuel cells, in electrochemical films, in electrochemical supercapacitors, and in dye-sensitized photocathodes. Nickel oxide acts as biosensor for the detection of hydrocolloid and visceral leishmaniasis. So nickel oxide has many applications. Now, what is cationic surfactant? I have told you one is cationic surfactant, N is positive, negative counter ions, anionic SDS, negative head group and positive counter ions. When the two are mixed, they form cationic. Sometimes what happens when cationic or anionic surfactants are mixed, that is, precipitate comes, little precipitate comes. Then what happens? Then you add more surfactants, both the surfactants, then the precipitates will be dissolved. And the surfactant is called cationic surfactant. Sometimes precipitates are formed, and you take the precipitate and dissolve it into organic solvent, then cationic surfactant. Now, synthesis procedure to form nickel oxide. What happens? We have taken two methods. In one beaker, we have taken CTAB only, and another beaker we have taken SDS only. In one beaker we have taken CTAB aqueous solution, and another beaker we have taken SDS aqueous solution. Now, so nickel oxalate, for the production of nickel oxalate, you choose two reactants. One is nickel salt, another is oxalate salt. So we dissolved nickel salts in SDS, nickel sulfate or nickel nitrate or nickel chloride in aqueous solution of SDS. And in another beaker, in CTB solutions, you add oxalate, sodium oxalate, oxalic acid, this. So here, within the code, oxalate ion will be there, and here nickel ion will be there. Now mix these two. Now mix these two, one beaker solutions added with another beaker solution, and shake, stir, and then nickel oxalate formed. Nickel oxalate formed, and these are nanosheets, nickel oxalate nanosheets. Their size has been determined by TEM. It is 180 to 230 nanometer nanosheets. Now the sheets are heated at 450 degrees centigrade in furnace. Then granular nickel oxide, blackish nickel oxides are produced. The size is 5 to 11 nanometer. Using cationic methods, we have produced nickel oxalate nanosheet and nickel oxide granular. And in method 2, that is, we first produced the cationic surfactant mix SDS and CTAB. Cationic surfactant is formed. You take it in two beakers. In both the beakers, material is same, cationic. In one beaker, you add oxalate. In another beaker, you add nickel salt, and then mix the two, and then produce nickel oxalate nanosheets. Here, diameter is length is a little bit bigger, 450 to 530 nanometer, and heated at 450 degrees centigrade, it will form hexagonal nickel oxide, nickel oxalate. When any oxide nanomaterials are formed, generally their oxalate are first produced, and from oxalate by heating, carbon dioxide goes out and the oxides are formed. So in this method, we have prepared nickel oxide. The nickel oxalate and nickel oxide which we have produced, they are XRD analysis. They are XRD analysis. From the XRD analysis, we have found different planes present in nickel oxalate and nickel oxide. Nickel oxalate 1, 2 and nickel oxide 1, 2 means prepared by method 1, prepared by method 2. These are the characterizations of the nickel oxalate and nickel oxide formed. This is the FSM and TEM analysis of the nickel oxalate and nickel oxide formed. Keep it in mind, depending on the cationic surfactant components composition, SDS and CTAB, their sizes differ. Nickel oxalate 1 by SDS CTAB ratio 60-40, 65-35 or 70-30. That is A, C and E. A, C and E, they are different. And similarly, nickel oxide produced by 60-40, 65-35 and 70-30, that is B, D and F. That is nickel oxide 1 and that is nickel oxide 2. That is nickel oxalate by method adapting method 2 and nickel oxide by adapting method 2. That is the depending on the composition ratio, SDS and CTAB, their structure differs. This is the morphology. Look here. When SDS CTAB ratio is 60-40 for nickel oxalate, it is nanoflex. Size, it is of nickel oxalate and it is of nickel oxide. And when 65-35, it is nanosheet. And when 70-30, nanosheet with layers and nanosheet with layers. Similarly, when SDS CTAB ratio 60-40, 65-35, 70-30, that is nickel oxide granular, and here granular, hexagonal, granular. So my point is here, it is tailor-made according to our choice, according to our reaction conditions, according to our ratio of the components, our desired product will be obtained. Next, HRTM analysis of the nickel oxide formed. That is set pattern, that is the distance between the planes, 111 plane of nickel oxide, 200 plane. The magnetic property, nickel oxide possesses magnetic property. So we also did squid-based magnetic study, which is anti-paramagnetic, which is paramagnetic. It has been done by squid. Actually, squid instrument is not available everywhere. In our time, it was available in DAE, Department of Atomic Energy. From there, we have done the magnetic property. Spin glass behavior showed for nickel oxide from by 22 to 27 nanometer size. Now, wastewater treatment. Look here. From wastewater, which contains phenol, we have taken added nickel oxide and light, that is photo decomposition. Light is irradiated. It is found, it is here only the wastewater, and there is no wastewater containing phenol, and there is no nickel oxide. So photoluminescence spectra, it is without any catalyst, A, and B, with nickel oxide 5 to 10 nanometer dimensions, and here C, nickel oxide with 22 to 27 nanometer. What we did, applications of nickel oxide, we take wastewater from industry which contains phenol. So we have taken some amount of wastewater and added some amount of nickel oxide produced by method 1, whose dimension is 5 to 10 nanometers. And in another beaker, we have taken the wastewater, added nickel oxide produced by method 2, whose dimension is 5 to 22 to 27 nanometers. And now what we did, we irradiated with light, with radiations, and after time 20 minutes, 40 minutes, 60 minutes, 80 minutes, 100 minutes, what we have seen, it is the peak of phenol that is decreases. That is phenol decomposed by the irradiation of light in presence of nickel oxide. Phenol is decomposed. Okay, then that means this nickel oxide prepared by these methods can be used for photocatalytic effect for the decomposition of phenol. So, and after one experiment, first experiment, what we did, we poured the water by filtering nickel oxide. That means that nickel oxide can again be used, and we have done it 4 times, and its activity remains almost same. That is, first time this, second time this with the same nickel oxide, third time this, fourth time this, then it remains same. Then measuring efficiency by this method, when we sent it to the publications, the reviewer asked us, in the market nickel oxide is available also, so your product is better than that, how will you prove it? So we again collect nickel oxide from market and did this experiment, that is in D, and that is decomposition occurs, but here efficiency is less compared to us. In our case, depending on size, activity changes. When the size is 5 to 10 nanometer, their activity was, efficiency was 81%, and here it was 79%. And in case of commercial nickel oxide, it was found it is 71%. So our product is more effective, more efficient. And then it was published in RSC Advances this time. So this is the photoluminescence spectra, plotted intensity, and E is the fluorescence intensity, and E is the intensity of residual phenol as a function of UV radiation. How much phenol is there after irradiation? It has been calculated, and if value of percentage of phenol degraded for each cycle having NiO as catalyst. We have prepared by cationic surfactant method. We have used another method, that is AFSA-80. AFSA-80 is a polymer gel mediated synthesis reaction rates. It is another soft chemical synthesis. AFSA-80 controlled synthesis or green synthesis of nanoparticles. So we had taken AFSA-80 and isopropyl myristate IPM and nickel ion, 1 nickel chloride, and in another beaker AFSA, IPM and oxalic acid, or sodium oxalate solutions. Then these are mixed, and then we get and then we got nickel oxalate with size this, and then after heating at 450 degrees centigrade, nickel oxide with size 20 to 30 nanometer. Okay. The product formed has been characterized by XRD and TGADTA characterization, that is its stability according to heat, according to exposure of time. We have calculated FESM and TEM analysis of the nickel oxalate and nickel oxide formed. This one. One important point is here, that is when some oxide nanoparticles are formed, first produce their oxalate, and after heating, then carbon dioxide will go off and we will get the oxide particles. The growth mechanism. When nickel oxalate is heated at 450 degrees centigrade, with different time we have collected nickel oxide, that is at 0 minute and 60 minutes, 120 minutes and 180 minutes. That is after heating, this time we got this type of nickel oxide. Again, their magnetic property has been measured by squid. Now, this is most important, antimicrobial activity. Antimicrobial activity of nickel oxide. That is, 1 gram negative and gram positive bacteria has been taken, and now what we did, we added nickel oxide solutions, added here, and it is found that nickel oxide is 80 microgram per milliliter is its MIC value, and 16 is MBC value. In case of P. aeruginosa bacteria, and in case of B. subtilis bacteria, here it is also MIC 8 and 32. We have calculated this. Look here. A and B. Fluorescence micrograph of P. aeruginosa gram negative bacteria. That is, we have taken the bacteria and we have taken its fluorescence spectra. That is, green fluorescence comes. Now treat with nickel oxide produced by these methods, and then take fluorescence spectra. This is like B. That is, bacteria are dead, the red lights comes. So this is the gram negative bacteria or gram positive bacteria. You have taken, take its fluorescence spectra, its green fluorescence comes. And now treat with nickel oxides. Then after some time take fluorescence spectra, and there is no green fluorescence, that means bacteria killed, and we get red spots. Now a dose, that is the effect of bacterial effect, antimicrobial effect studied. And now question is, it is harmful for human blood. Now what we did, effect of nickel oxide on human blood. Control is used as one of the detergent. Generally, CTAB or SDS is used as positive control. And nickel oxide, if you add 50 grams, no effect. 75 grams, minimum. That is, with addition of microgram per milliliter, the hemolysis, human effect on human blood is like this. That is dose response plot for the hemolysis of the human red blood cells in presence of nickel oxide nanoparticles. In hemolysis, as a Tritonix used as positive control, and red blood cells with different amount of nickel oxide nanoparticles. This is the antimicrobial effect of nickel oxide. That is the effect of bacteria cells, fate of bacteria cells in presence of nickel oxide. These are the bacteria. One is gram positive, another is gram negative. Add nickel oxide, the bacteria are killed. It is the mechanism of antimicrobial effect of it. There is another method. I think time is short, so I shall make it okay. Synthesis of europium zero nanoparticles by gamma radiolysis. I told you that three methods. One is reverse micellar method, another is cationic surfactant method, another is radiolysis method. What we did actually in a DST meeting, I was challenged that to produce europium zero nanoparticles. They said that you will not be able to produce it. I said that I must do. Then what we did, europium nitrate, polyvinyl alcohol and polyvinyl phenol, and isopropanol, CH3 whole to CHOH. We taken aqueous solutions of these two in a container, generally a large beaker, and then gamma 60 radiations is irradiated. 4.6 is kGy per hour, 1.35 millimolar solvated electrons per hour is produced, and this isopropanol is added for radical scavenger. If any radicals are found there, they will be destroyed. Environmentally, single step. What happens? That is the gamma radiations from 60 cobalt in the solvent molecule. These solutions, it is added, and solvated electrons are produced. They will reduce. That is, what is this? H2O, gamma radiolysis is produced, solvated electron. This electron, metal ion, europium plus 3, it becomes europium 0. Why? Controlled reduction of metal ions only by the solvated electrons and secondary radicals instead of introducing any other interfering entity into the synthetic mixtures. Reactions can be carried out at room temperature, only mixture and take irradiations. The hydrated electrons seem to be strongest reducing agent. The electrode potential is minus 2.87 volt against normal hydrogen electrode, and H plus H is minus 2.3 volt normal hydrogen electrode. So it is better. It can reduce. So europium 0 is produced. How will you know? Take its absorption spectra. This is the absorption spectra. When PVP has been used, when PVA has been used, that is this is the absorption peak of europium. Absorption and emission spectrum of europium 0. This is the absorption spectrum of europium 0. This is the absorption spectrum of europium 0, nearly 275 nanometer. And this is the emission spectra, illumination spectra of europium 0. That is near 380 nanometer. That is this. When PVA has been used and when PVP has been used. Mechanism of the reduction. I have told you solvated electron. That is the electron, solvated electron, it produced europium 0. And the particle size formed, it is like when concentration of europium differs, size also differs from nearly spherical to spherical elongated. This is 10 pictures of europium nanoparticles. Irradiation dose 83.88 kJ. This is the europium 0 nanoparticles, black particles. Their FTIR analysis bonds, the frequency corresponds to the europium zero bonds. This is the conclusion. What we said, the large here it is gist. First work underlines the efficient synthesis of highly crystalline, monoclinic, rod-shaped and flower-like rare earth phosphates, lanthanum, employing reverse micellase soft template at room temperature. Change of reaction condition causes variation in nanorods dimension as well as formation of nanoflower. Synthesized nanorods are sufficiently luminescent with high quantum yield, and energy transfer from cerium ion to terbium ion occurred via multipolar interaction, dipolar quadrupolar interaction, explained by Dexter mechanism, and the distance between the two ions 11.4 angstrom. Electron transfer process also confirmed through time-resolved quenching of cerium lifetime. So next we designed a soft and facile cationic surfactant controlled approach for preparation of various nickel oxalate nanostructures, including single-layered nanosheets, multi-layered nanosheets, and nanoflakes. In short, we developed a simple, controllable, and environment-friendly bottom-up pathway for synthesizing nickel oxide nanoparticles, which are characterized by means of XRD, FESM, and TEM. Magnetic study of nickel oxide with size 5 to 10 nanometers so canted antiferromagnetism, but for a 22 to 27 nanometer grain size produced by method 2, we find surface spin disorder resulting in spin glass behavior. Magnetic study indicated their spin glass behavior. The as obtained nickel oxide nanostructures show high photocatalytic activity to decompose phenol dissolved in water, displaying great potential in the application of environment pollutant cleanup. I have told you for diverse applications. The broad spectrum of antimicrobial activity of these nickel oxide nanoparticles with low emissive values clearly indicated their efficiency towards inhibiting the growth of gram-positive and gram-negative bacteria. They are killed. And 10 pictures of bacteria treated with nickel oxide nanoparticles indicated that the antimicrobial action was governed by membrane disruption, you have seen in the figure, and cell lysis, and maybe stable positive surface charge played a vital role in it. Their low hemolytic activity at bactericidal concentration further makes them useful for biomedical applications. For its easy preparation, low cost, high abundance, and excellent bactericidal effect, nickel oxide has a promising role for future antibacterial agent. Then we did radiolysis reduction of europium 3 plus ions, it's zero valent pure europium nanoparticles. From europium 3 nanoparticles, by radiolysis, we produced europium 0 nanoparticles. This method is a single step one and chemically less hazardous. The radiolytic synthesis may be considered as greener one since reaction was carried out in water in presence of PV and PVP as stabilizer. The observed optical characteristics can generate more interest in future to investigate the transition behavior of the europium 0 nanoparticles, magnetic nanoparticles in the and also europium 0 metal nanoparticles in the solution phase, which may have ramifications applications in the field of optoelectronics. Now I think our time is up.
Thank you, sir. Now this session is open for discussion. If anyone want to ask any question, you can unmute yourself or post your question in the chat box.
Yes,
One
yes.
question is there in the chat box, sir,
Mhm.
from Dr. Namrata Roy. What is the reason behind change from nanoflakes to nanoseeds on changing the composition of the cationic surfactant in case of nickel oxide? Does it
Yes.
depend? Okay. Another
I,
part is there. Okay, okay, you carry on.
yes, actually, I told you that reaction depending on reaction conditions and the ratio, because size changes, size of the core changes, that depending on the size, the size of the nanoparticles changes, either spherical or granular or seed shaped. That depends on the uh, size of the core.
Okay, it has another part. Does it depend on the size of the reactive core which is formed by the
It's
surface?
sir just confirmed, so it's fine.
Confirm,
Sir
sir.
just said the
Yes,
same
okay.
thing. Yeah.
Okay, okay.
Okay,
Yes.
thank you, sir.
Okay. Anything more?
Any more question? Okay, another question is from Dr. Kaushik Sarkar. Nickel oxide produce mitochondrial ROS generation as per knowledge. That's why nickel oxide has any type of effect on blood cell.
Nickel oxide has effect on blood cells depending on its amount. I have told the figure. In the figure, it is found that up to 250 microgram effect is minimum. We have analyzed it. We have
Okay. Okay,
uh,
sir. Up to 250 milligram, it is safe.
not
That's
milligram, not milligram, microgram.
microgram, microgram, sir.
It is safe.
Sorry.
It
Okay,
is safe.
okay. Okay, sir.
Okay, any more question? Okay, no, I think no more question is there. So I sincerely thank Professor Bhattacharya for an enlightening talk. Your discussion on soft chemical clearly highlighted how this method help introduce ligands, prevent particle aggregation, and control the shape and size of the nanomaterial through factors like solvent, temperature, and reaction time. We appreciate your insights into the advantage of short routes, low temperature, simple apparatus, low cost, large scale potential, and versatile post treatment. The examples showing nanorod and nanoflower and characterization by time analysis, lifetime analysis, efficient fluorescence spectroscopy, etc., are also very informative. Your explanation on the cationic surfactant control approach and their application are highly inspiring. Thank you, sir, once again for sharing your expertise with us. Thank you, sir.
Thank you. Thank you all. So I am leaving now.
Yes, sir. Yes.
Okay, thank you. Thank you.
As I can see, our next speaker has already joined. Dr. Dipendu Das, are you there? Dr. Das? Dr. Dipendu Das, are you there? Dr. Das, am I audible? Dr. Dipendu Das, am I audible? Yes. Okay. Visible now. May I proceed? Dr. Das, please unmute yourself.
Yeah, I just joined. I was just stuck in a meeting. I'm sorry.
Okay,
I
no
just
issue,
turned on
no issue.
my audio. So shall I start presenting? And
Okay, let me introduce you.
sure.
Okay. So I am delighted to introduce Dr. Dipendu Das, Associate Professor of Chemical Science, ISER, Kolkata, West Bengal. Dr. Das earned his PhD from Indian Association of the Cultivation of Science India, and completed his postdoctoral training from Emory University, USA. His research group focuses on the emerging field of systems chemistry, chemical evolution, and peptide nanotechnology that are reshaping our understanding of molecular interaction and cell assembly process. Dr. Das has received numerous prestigious awards and distinctions. Notably, he has been honored with Vigyan Yuva Shanti Swarup Bhatnagar Award in Chemistry in 2025 for his pioneering Life 2.0 work on creating lifelike system from simple chemical building blocks and exploring the chemical origin of life. It's really a great pleasure for us to have such eminent personality with us today. He also received Swarna Jayanti Fellowship in 2020 and selected as a Indian National Scientific Academy Associate Fellow in 2025. He won the Indian Peptide Society Young Scientist Award in 2021 and CRSI Bronze Medal in 2023. His achievements are highlighted in the 75 under 50 scientists shaping today's India compendium. Internationally, Dr. Das is widely recognized. He has been elected chair of the Gordon Research Conference of the System Chemistry 2028 and as vice chair in 2026. He has mentored Gordon research seminars. He has published his work extensively in leading journals such as Angiochem, JAX, Nature Review Chemistry, Nature Communication, Proceedings in National Academy of Science, etc. He has represented India as a at global forums, including the Second Commonwealth Chemistry Conference, and served on advisory boards of Asia's Chemical Reviews, Material Horizons, Chemistry System Chemistry, RSC Organic Chemistry Frontiers, and Asian Journal of Chemistry. So we all are excited to hear from you more, sir, regarding your research and contribution today. Now this online platform is yours, sir. Welcome you again.
Okay, thank you. Thank you for the introduction. I didn't catch your name, sorry. You are from Maulana Azad College, right?
Yes, yes,
Yeah.
yes.
Okay, okay, thank you. I will start my talk. Just two quick points. One is that if you have any question in between, or taking question in between, you can say raise the hand. All right. Okay,
Okay, sir.
and sometimes my audio goes blank, so I can keep on going, but I might not hear your question. So for that, you have to again raise your hand. All right. Yeah. So I'll start the
Okay, sir.
presentation. You can see the slides, I guess, right?
Yes.
Okay, all
Yes,
right.
sir.
So
Yes.
what we work on is a rather very new field of chemistry, that is systems chemistry. So let me just, yes. So I will talk about that, and it's a very provocative title in the sense that network of reactions are used. So let me just, one second. There's a problem with the screen. View, one second. Sorry for the delay here. Now you can see hopefully again. And yeah, so systems chemistry deals with to the fundamental question that one can ask, human being can ask, that is how life started on this planet Earth, and how we can create living matter on the bench. This is something that has been exciting to, I think, all scientists and all human beings, because it's a cognition problem, right? How life started and how life originated on Earth, and an equally tantalizing question of whether we can create life on the bench. I will start my talk with a very interesting quote by theoretical physicists. Do you believe? Usually, physicists talk about big problems in the context of astrophysics or condensed matter or something, right? But not life. So, Edwin Schrodinger, as you might know, many of you, all of you might know him. He was a quantum uh, theoretical physicist. He said a very nice, he wrote a book on living matter, and wrote that how living matter evades a decay to equilibrium. He defined in a way, and obviously it's paraphrased because he wrote in German or Austrian. He was an Austrian scientist, but It's very powerful term when you see the statement, living matter evades a decay to equilibrium. And throughout this talk, I will try to give you the idea that what do I mean and what does it mean by that, and whether we can create chemistry or chemical tools to create living matter-like objects. So it's a very interdisciplinary field, the systems chemistry field. It is, you can imagine the interdisciplinarity from a court of physicists about living matter in a chemistry talk. So that interdisciplinary the field of system chemistry is, and it can be shown in a very rather abstract art where multiple reactions are going on. A bunch of reactions are going on. Small molecules are interacting with each other and can have hierarchical assembly, not necessarily under equilibrium. So I keep talking about equilibrium, and what do I mean by that? It will be slowly clear, hopefully, in the throughout the talk. So then, under a pressure, a selection pressure, one of the components really grow and outcompete others. They proliferate more. They are more forming more. If I say in the synthesis or synthetic organic chemistry terminology, they form more over the other. But whenever we turn off the energy source, they come back to the equilibrium or the same ratio as the ring sizes are sort of mentioning. So these are very abstract. I'll show you the structures, the very simple structures, and I hope you appreciate that. And that is really connected in the root of it, as you by now might have guessed, uh, origin of life, right? If you can understand how small molecules start interacting to form really complex structures, polymers, and complex functions, then in principle we can also answer questions of chemical origin of life. So we have two big goal, two very exciting goal that should relate to all human beings. One is how chemicals evolve to living matter. It's a 3.8 billion years old journey, so we might not ever know how it happened, but some ideas can be there. And equally, at the same breath, we will talk about we work on whether we can create life like objects, life 2.0 on the bench. All right. Okay. So and based on that, what is the meaning of life 2.0? What exactly lifelike objects are there? I will show you that. And what is the use of that? Let me close the window. There's too much of sound from outside. So what is the meaning of that, if I have to say that, and what should be the usefulness? And that also is going to be in I'll show you some snippets on that, a couple of slides, few slides on that, what exactly the function can be understood from these two fundamental questions. Okay, one is how life originated, and second, how whether we can create life 2.0 on the bench. All right, so create life on the bench, and we publish this very provocative review. You may have a look at it. If you don't have access, you can send an email to me. I will be happy to share the paper with you. This is a perspective very highly cited already, but this has a section which talks about how one can create actually living matter on the bench. Okay, and it's outside of the chemistry that we are known. It's not necessarily has to work with nucleobase, peptides, and sugars. It can be any molecules, simple enough, but they can show certain traits of living matter. And what are the traits? These are the three gears I have shown here. Three gears. As you know, cycles have gears, right? All engineering logos have gears, right? Gears are very important. They have to move together to create life. In this context, I have shown three gray gears. These gears have, one is called replication, very important. For living matter to be created, replication should be there. There is information transfer. Basically, one information has to be transferred from one side to other. Or mother to offspring. Second is reaction network. How metabolic reaction network or reaction network can lead to metabolism. By metabolism, I mean, let's say, a degradation of molecule leading to energy release, and that energy is helping us talk, move around. Very important. And finally, compartmentalization. We are all a bag of polymers, right, in a way, but we have a skin, right? We have a compartment, we have cells, we have everything. Is even the simplest life forms will have a compartment or confinement around it, right? So these three check boxes need to be ticked, and we have talked about that in this review. And more importantly, one gear is missing, and that's where the fundamental point of Schrodinger also comes in, that is out of equilibrium. Now, out of equilibrium, all have to be out of equilibrium, because the moment we reach equilibrium, we are in death phase. Anything that is in equilibrium is actually not living, right? This this mug of water, it's in equilibrium. It's not living matter. But if you take a solution or a liquid, let's say from a water side, even a water pool is being gathered after a bit of rain, you see under a microscope, they are living life forms, right? So anything under equilibrium is not living. So this review basically ties up these three concepts or four concepts together, although four gears are not easy to move, as you can understand, they will be short circuiting. So one gear has to be offset. That is what we're going to find out. But these four gears, these four check boxes need to be ticked if before we begin, or rather we begin our journey to ask questions of living matter like properties of light 2.0 on the from the chemistry bench. And when we do that, we get really complex functions. For example, oscillations. Oscillations is something that is very rooted in biology. All of the systems are in oscillating mode. We are in a systems level oscillating also, where we have sleep, wake cycles. We have this heating homeostasis that is in a way in a non-steady state. We have autopoiesis. What is autopoiesis? It means self-synthesizing. Maybe I'll show you a few slides on that. And motility, I will just show a snippet on that. Not more than this. These 2-3 points I will show because of the constraint of the time of the talk. And from there, we will see whether we can create, although this is too far now, we are miles away from this, maybe next 20-30 years, at least whether we can see when we create this, these check boxes are ticked, whether we can begin unraveling concepts of oscillation, autopoiesis, motility, and organization or non-equilibrium organization, very, very complex behavior, typically reserved for biological living systems. Okay, so yes, and as I said, the moment we do that, can we use those know-how, we use those understanding skill set to create objects or materials that are really adaptive and active and can serve a function for humankind, right? Okay, so when you think about biology and these videos keep popping up, biology has exotic structure, right? For example, these green filaments that are forming and breaking as a function of time, these are cell division, as you may know. This is seen across life form as an evolutionary strategy. You have to divide, right? You have to grow, and this is using GTP. Now here is the chemistry part. Slowly we'll enter chemistry. This is using GTP, a guanosine triphosphate, or NTP per se, nucleoside triphosphate, that breaks down to NDP and phosphate, and a lot of energy. This energy is used to form this structure that is ripping apart to daughter cell, forming this kinetochores, right? Similarly, this alien looking guy is trying to evade or catch this foreign body. This is basically WBC, is trying to catch it, and it's also using NTPs, again nucleoside triphosphate, which are having this high energy molecule, which are basically thermodynamically activated, kinetically stable molecules. Very powerful words. Thermodynamically activated, but kinetically stable. They are kinetically persistent, but the moment they hydrolyze, they release a lot of energy, right? Similarly, the sugar crystal will be hounded in a moment by bacteria, all using energy sources. As I said, in a systems level, we feel cold, we shiver, we feel hot, we sweat. It's also done by these non-equilibrium processes, right? They're all engraved in biology. Biology is not in equilibrium. Okay, I'll show you a very simple diagram to maybe connect the dots. What do I mean exactly by equilibrium and non-equilibrium? These are used, these are driven by chemical fuels. What is a chemical fuel again? Chemical fuel is an NTP or a GTP, ATP, which is a thermodynamically activated molecule but kinetically stable molecule, which when broken can release and form phosphate and ADP or GDP. That's okay, but a lot of energy. That energy drives these structures. That leads to let's say I'm lifting a weight. That is also ATP driven, right? There's some warm heat generation in my hands, right? Because that's heat is being dissipated, and the heat comes from the hydrolysis of the fuel, the currency of biological system, that is ATP. So if we can actually make this system, and if there's a physicist out there in the audience, I don't know, there might be a physicist, for them, this will be a non-equilibrium system, right? They're all non-equilibrium system. They're not equilibrium, right? They're all transient systems. So this will be a non-equilibrium system where you can say that this is also energy driven, right? You have to have energy of the pump to maintain the flow of the fountain. And similarly, just like all of us, this guy on the screen is also a non-equilibrium object. So we want to create non-equilibrium systems on the chemistry bench, ideally using very simple molecules, and whether we can access really complex behavior. That's the fundamental question. Can we create life-like objects, living matter-like objects? And second is, can we use them for functions? Okay, that was the point. And again, I think you might have seen this video also. This is also a directional motion of a protein, a walker protein, that is burning ATP to ADP, is called power strokes, or sometimes called this non-equilibrium kinetic symmetry is going on, right? These are not going up and down, they're going only in one direction, and that is inherently non-equilibrium, right? Okay, so this is also using this very childish cartoon to show that you add the fuel of ATP and water, ADP and phosphate is releasing, but very importantly that we usually don't count a lot of energy is released. These energies are leading to this directional motion, okay? And that leads to also, as I said in the earlier slide, chemotaxis. I will skip this, but just like chemotaxis is also a very interesting part, right? We smell a food, we go to the kitchen. I'll come back to that maybe in just a bit, but that is also energy driven processes. All right, so if I define that, and for a physical chemist out there in the audience, a very simplistic diagram, if I try to define what is equilibrium and what is non-equilibrium, because equilibrium has been taught, but non-equilibrium is not taught per se in a normal curriculum of a chemistry book. So equilibrium is, very simply put, you don't need to add matter or energy to sustain a state. In other words, you don't see a change on a reasonable amount of time. Let's say you have a bottle of water and you don't see any change in the in the system as a function of time, and you don't need to add energy or matter to sustain the state. So in this case, this will be under equilibrium. In COVID time, we used to wash hands, and maybe COVID is the reason why we are meeting online also. This online was facilitated, but anyway, COVID time we are using washing hand to form micelles. Micelles are in a way in a equilibrium state or in a kinetic trap state. You don't need to add energy or matter to sustain the self-assembly. So what will be non-equilibrium? Non-equilibrium will be the simple similar definition will come in. You need to add energy or matter to sustain the state. A non-equilibrium state will be you need to add energy or matter to sustain the state. And if I have to draw the diagram, and diagram can be a bit open to debate, and that will be nice to get questions on that. This will be the diagram where you have building blocks which are not aggregating, but you add an energy source and they start aggregating. You add light, they start aggregating. These photoswitchable ligands, for example, if you can imagine that, they start aggregating, you turn off light, they come back to equilibrium. It's like a balloon which is descending on the ground. Let's say a balloon, air balloon, slowly descending on the ground, but you are gently putting air or blowing air to that. It will be remaining in the air as long as the source of air is there. But the moment you turn off the air, it will descend down to the ground, right? So the air was being added in a way energy source to sustain this state. You can imagine a very heavy ball you're trying to hold in a mountain slide, or maybe somewhere in the mountains in Sikkim or somewhere. You're holding it because you're adding the energy. The moment you let it go, it will go down, right? So this will be a non-equilibrium steady state if I keep adding energy. This system is sustained there. And that is very, very important again. So the simple definition is this. In this case, in equilibrium case, it will be a downhill event, or this state is achieved. You don't need to add matter or energy to sustain the state. And non-equilibrium will be you need to add matter or energy to sustain the state. And the state is there. This will be non-equilibrium steady state. In principle, you can see that system to be static. For example, a refrigerator is in a way non-equilibrium steady state, right? Our outside temperature can be 40, but refrigerator can be as low as -4, right? So why? Because somebody is paying the bill. So who is paying the bill? Electricity, right? Energy is being added. In this case, who is paying the bill to go up? The chemical fuel. What is a chemical fuel? A thermodynamically activated kinetically stable molecule, which when hydrolyzed or degraded can lead to waste product. Fair enough, that happens. A plus B, or A to B and C. But A to B and C plus energy, that energy is used to access these structures. Okay, and hence these active polymers of biology can be seen. The structure I showed you earlier, the quintessential polymers of biology that rips apart cell, that helps in movement of WBC, that helps us lift a weight, that is always happening in the body, right? Those are using in GTP. They are all non-equilibrium. This is the quality of the statement of Schrodinger will be anything that is under equilibrium will be in death phase. And what is and living matter, and hence the quote comes in, living matter will evade the decay to equilibrium. We'll all try to eat our food more or less on time to sustain or live happily, right? We stop eating for 1 week, 1 month, we'll die. So it's very, very important to use the energy of the molecules that is being released, and that energy, after degradation, that energy is used to fuel our body, because we are in a non-equilibrium steady state, or rather non-equilibrium state, right? Away from equilibrium. All right, so that is the fundamental concept. So when we looked at the structure, and I'll go back now in a bit more detail of the chemistry, you see this video, these polymers are forming. These are very inherently, if I deconstruct them and make it very simplified, this can be taken as a protein, a building block. It is there, let's say tubulin. In presence of GTP, let's say this is GTP, they start forming this polymer that you can see. Interestingly, this polymer starts breaking the GTP to GTP. And phosphate, you can see phosphate here, GTP here, and it starts breaking. Okay, so the fact that the GTP led to substrate, GTP is a substrate in a way, GTP drives the polymers of this building block, this lag here, building block to form these polymers. Now, the polymers subsequently after formation start breaking the GTP. So then they come back to, again, the polymers or the building blocks, that is the tubulin, this is the microtubules, the green filaments, and they come back to the building block. What is happening in the process? GTP is forming phosphate and GTP, and the energy released from this hydrolysis is leading to the structures formation. At one point, the structures are stabilized by GDP. These polymers are stabilized by GDP. However, if you take a separate experiment, add this protein, this building block, this tubulin protein, and add GDP and phosphate, you don't see ever polymerization. So for polymer formation and for dynamic instability or dynamic structures like this, you have to couple the process of GTP to GDP plus phosphate plus energy with this process to get this structures formation. So these are very, very fascinating ideas that we were exposed, and this very old field though, last 30, 40 years of intense research has gone on, particularly in biology. But we really love the concept, and we thought there are two critical parts here, if you notice. One is this protein building block can break GTP actually, but in a very low rate. However, once the structures are formed, they break GTP in a very accelerated rate. The rate of hydrolysis of GTP to GTP is extremely high. And that leads to, in a way, a negative feedback. Okay? So it's like driving a car. If you drive the car too fast, you burn fuel faster, right? And you will end up, so burning fire. So it's like a negative feedback process. Faster doesn't mean that. So here the GTP is leading to the polymers. The polymers subsequently break the GTP, and the negative feedback kicks in and comes back to the equilibrium state. So that was very interesting. We found out two things here. As I said, this can break GTP, but this breaks GTP far faster than the building block. All right. So that was the take-home message, and and a lot of energy is released. These are not our papers, but they show that these polymers, when they break apart, rip apart, they have really a ton of force generated. So we started with a very simple molecule published in 2022, a bit older paper. In fact, earlier than this, we have started the work. So this peptide we took, the idea was to add a chemical fuel, which is again thermodynamically activated, kinetically stable molecule. We did not take GTP because GTP is a bit too kinetically stable, very, very strong bond to break, right? We took a cousin of that is a carboxylate ester. All right. When you mix them together, we use this imine bond. So imine, once they form, this forms this. The amine forms this aldehyde to form an imine, and these peptides are formed. Okay, there's a lag here. Sorry. So yeah. Anyway, this peptide imine is forming. This is a very standard textbook organic chemistry reaction. but this is not usually feasible in water. This is largely water medium. So how it goes on? Because the next step, once it forms this imine, they start assembling. They start assembling, they start aggregating, and once they start aggregating, they are going out of the system. It's like precipitation of our equilibrium product. So what happens? They go out of the equation, and hence the equilibrium shifts this direction, and it keeps on going this direction. Okay, from here to here to here, from here to here to here, and hence it's a positive feedback in a way, right? The self-assembly helps self-templating the same reaction. I hope you're understanding this bit. Okay, again I will repeat, it forms an amine, aldehyde imine, usually not very stable in water because it's very reversible. Water is generated as a side product. However, if these imines start assembling, it will form these structures. Now these structures being structures not in soluble phase, this leads to more and more of the right hand side going in, the equilibrium have shifted, and that leads to an positive feedback. So more and more shifting happens from left to right. Okay, now what happens after that? We have an activated molecule, right? As you know, this is an activated molecule, is an ester molecule here. This is an ester molecule here. We see actually a physical hydrogel forming, but in the hydrogel, they are very close to each other. Now, what happens once they're very close to each other? The imidazole starts breaking the ester a bit faster. Just like what happened in the microtubules, the tubulin was able to break the GTP, but the microtubules, the polymers, broke the GTP faster. There's a reason to that. I will not go in depth of that. But here, because of the histidine to be very close, the imidazoles are very close. They are, if you can see my video feed, they are very close. They are doing this cooperative catalysis. Maybe I'll detail it out in case somebody has a question on cooperative catalysis. But this forms faster rate of hydrolysis of the ester, and that leads to subsequent degradation, because once the ester is broken to acid and alcohol, as you can see here, okay, these two, right? So this leads to breaking of the gel or the structures as a function of time. We see sol, gel, sol, or rather no fibrous assembly as you can see from the TEM, and again no assembly, literally less assembly as a function of time. We could see from my rheological experiment, that is a mechanical property measurement, is an odd, this is a y-axis in log scale. We see orders of magnitude more strength in the gel vis-a-vis the control. The interesting bit is that this lysine plays a very important role. This lysine, the primary amines, play a very important role. It's like opening a bottle of, let's say, a water or these nuts, let's say, right? This cashew nuts. If I have to open the bottle, I have to hold it with my left hand, right? If you can see my video feed, and the right hand will be the guy or the molecular unit that is doing the actual job. However, if I don't have the left hand, which doesn't look like doing a very important job, but it's very important job actually, right? Holding the container, the right hand is the catalytic unit. You can imagine on the screen we have a imidazole, which is the right hand here, and the left hand is the lysine, which is doing a reversible imine bond formation. So one reversible bond formation is helping in anchoring the substrate, and the other imidazole is helping in breaking the substrate, right? So it's very interesting in the context that it is showing a catch and break scenario, right? So what we did when we did a control, when we mutated the lysine with an arginine with guanidine group, is similar pK, or the lysine has 9-ish, it has around 11 pK, it didn't show any rates that much. However, when the lysine was there, it was show substantially higher rate, and these transients, structures, formation and breaking could be seen. In fact, this concept we have shown earlier also in 2022 and last year as well. I will not talk about those in detail, but essentially what we are trying to say is that a substrate driven self assembly is happening, and maybe in the next slide I will show that a substrate driven self assembly is happening. If I compare and juxtapose the cytoskeleton polymers here, these are the biology event. These are very complex, seen across life forms as a evolutionary strategy, very complex systems. Here also fundamentally same thing is happening. Here the protein building block is driven by GTP to form these fibers. In this case, these peptides are driven by this carboxylate ester to form this fiber, the fiber subsequently breaks the carboxylate ester. In this case, these fibers breaks the GTP to GDP and phosphate and comes back to equilibrium, right? The building block. We can refuel them just like you refuel your car, you refuel yourself with a beverage or a food packet, right? This can be refueled by addition of one more batch of fuel, which is basically thermodynamically activated kinetically stable molecules which can drive this process and subsequently be degraded. This concept, this very concept, this substrate driven self assembly and subsequently broken to coming back to equilibrium, the sol, gel, sol, or phi, or no assembly, self assembly, no assembly concept has been shown by us for the last many years. All these concepts, all these papers have the same concept, and I don't have any shame in saying that because it's to just to generalize the fact that substrate driven self assembly and catalytic self assembly that can subsequently break the same substrate can lead to non equilibrium structures, right? Structures forming and breaking as a function of time. So far so good, so we were happy about that, but there was always this feeling that why we are not seeing those transient structure, that is this biological cytoskeleton proteins are showing that as they are forming and breaking away, because we never had a confocal. But then thanks to Swarna Jayanti grant, what the introducer mentioned earlier, that we got a confocal in the lab and we could see that indeed this system shows this dynamic instability in a way. The structures are forming and literally dissolving the next one in front of our eyes. We'll see some of them are dissolving here. If you notice, some of them are dissolving. We can see from the TD channel or from the phase contrast microscopy, the same image, you see that they are dissolving. We can in fact see even complex behavior like treadmilling. I will not go in detail because we are also learning this. This is a bit biological or biology concept or microscopic concept where monomers, they are shown to be going up and going down, or maybe they are just coming together and then they're vanishing literally. Rather, vanishing is not a right scientific term. They're dissolving immediately, showing catastrophe. Catastrophe again is a terminology used in cytoskeleton polymers. It is showing treadmilling, just like we do treadmilling. They can form one side and break from other side. We can see from the video also. I will run the video again. You can see that some of them are forming and going away as a function of time, as you can see that. This was very telltale sign that these are forming and showing complex behavior just as we see in biological systems. Okay, so these are a coupling, you can say, of catalysis. Catalysis plays a crucial role here. It's coupling of catalysis, coupling of non-equilibrium systems, coupling of metabolism in a way, right? We are using these esters as in a way food to do these reactions. Next was this work we did try to do an oscillating system, because oscillation is something that is very remarkably engraved in all living systems. Oscillation is very unique also. I don't know how many of you have heard the experiment called Belousov-Zabotinsky reaction, where a beaker changes color as a function of time, red, blue, transparent, red, blue, transparent, or many colors could be there, and that is oscillating, right? So we wanted to see whether oscillation can be shown in these systems. So what we used a remarkably simple molecule that you can purchase from anywhere, right? Only a histidine, and we used a substrate, this ester. When we used A plus BC, it forms ABC and forms a gel and forms this fiber. By now, you might have guessed these fibers will subsequently break the ester. There will be negative feedback. The negative feedback kicks in, leading to acid and alcohol, and a salt state is formed. However, when we did not add the buffer, and it's very interesting part, when we did not do this system in presence of buffer, rather we did the system in absence of buffer, there what happens, the pH of the medium, actually I'm rushing through this because I presented this earlier also, some of you might have heard this in some platforms, but anyway, the fundamental concept is very, very basic. What happens if I don't use a buffer, this acid starts promoting the same reaction via aminal formation, hemiaminal formation. Again, a very textbook reaction, BSc first year reaction, carbonyl chemistry, where acidity leads to equilibrium shift from left to right. Imine formations are better when we have acidic condition, right? Here also, when the acidity, we start from pH 8, the acid the pH drops down to 7-ish, and that leads to more and more of this equilibrium from left to right, and that leads to again a gelation formation, and again this happens as soon as gel forms, the negative feedback that kicks in, and that leads to again a cycle of sol gel, sol gel. We could see this multiple cycle of sol gel, multiple cycle of sol gel as a function of time. Okay, so now in the next part of my talk, I will talk about compartmentalization. That is something that can be exciting to many of you, because we all, yeah. So the point is that compartmentalization is something that very exciting to us, right? We see these liposomes, vesicles. Liposomes are one of the first marketed nanotechnology product that can be used as a drug, right? Liposomal solutions are used. Many of you might be working on the field also. So, yeah, there's some. One second, let me just. My audio is getting. Okay, so maybe I don't know. I have to see that. Yeah. Okay. So next would be compartment. Can we couple compartmentalization with life death events? Can we show compartments that grow and then possibly divide and possibly die when the food is not there? Can they show living matter like behavior? And not only vesicles, can we show these condensates? Again, is a very new topic. Biomolecular condensate. Many people call it as liquid phase separation, co-sorbate also many people call them. It's a very interesting topic, and it's a newly new terrain of research. In case you have not read about it, I will talk about this story in just a bit, but I really suggest to read it, because it really has relevance from human health to origin of life, right? It's very profound concept. Okay, so I'll talk about first these vesicles. We use the same concept, exactly same concept, published this year in Chem Journal from Cell Press. What we used the peptide with amine. As you know, amine is the left hand, right, holding the bottle or the can, and the right hand is the histidine, right? So we use the same concept in an attempt to find whether vesicles can be formed or not. Indeed, when we added them, they were forming rapidly imine formation. We suggested that indeed there could be some self-assembly process that is leading to left to right movement. We can see turbidity coming up as a function of time. There is a parallel turbidity, y-axis. The y-axis values are parallel to x-axis, which is quite interesting. Suggests that might be a pseudo non-equilibrium steady state, where the formation and breaking of the structures are happening at almost the same rate. Okay, and we in the microscope we could see indeed structures that are very spherical, from cryo-EM to optical to confocal microscope. You can see them to be forming rings, and when we do this line analysis, they are forming these two nice little peaks, which suggests that the dyes are really bound on the surface of the membranes. Okay, so yeah, so we characterized with a lot of NMR, and that was interesting to see that indeed these are forming, the means are forming. But this video will run now. We did XRD and also from DFT analysis from Lisa Roy, IIT Kharagpur. But this video will show now. Initially, everything is soluble. These two are soluble. Okay, now we see that they start forming this structures which look like living matter. Slowly some vesicle like structures are forming, and because of these structures, you can see from the skin, little hand, they're forming these vesicle like structures, and they're forming this oblate porate shape. Fair enough, but they're forming this rather homogeneous structure. And I'm not running this video for long enough, but if I do run it for long enough, you will see that.
Sir, can I ask a question?
Yeah, sure.
Sir, this spectra has been recorded on semi-solid sample or liquid sample?
Liquid sample, confocal liquid sample.
Okay, because if sample is gel-like, then it becomes difficult to record solid-state NMR sample.
No, this is NMR. NMR is for liquid, liquid state NMR. This is NMR. Yes, there's some peak broadening, but not too much.
Yes, sir. Because as it will turn towards gel state, it will be difficult to get solution state.
Yes, difficult, but sometimes you can get some peaks which are very broadened. It's very difficult
Light
to quantify.
broadening will be there. True, sir. True, sir.
Yeah.
Thank you.
Yeah. So this is what is happening as a function of time. They're forming from literally blank confocal image space to structures that are very spherical and rather vesicular-like structures. And importantly, after some time, if you run it for long enough, there will be nothing left. They are death phase. Okay, so it's showing growth, death phase as a function of time. Interestingly, what we did was this. This is again a very simple molecule we added. Now, this is the reaction cycle. I will repeat and run you through again with this cycle. With peptide, this dipeptide basically, and this E1 ester, when we added them together, this form this imine and this amplify. This amplify starts aggregating, and then this aggregated structures form these images, and then they come back to equilibrium, and when it's formed this P again, and some this is lag. This acid and acid aldehyde or alcohol aldehyde, and this acid is forming, right? Because this is getting broken, right? These two are forming. At this stage, we thought that if we again don't buffer it, so what will happen? And if I add, and if we add something like this molecule, and this is again an acetal, acyclic acetal, which is very labile to acidity. The idea is that this will be initially there as a spectator. E4 will be there, let's say. E4 and P will not do anything together because they don't cross talk, right? There is no aldehyde here. If I have a pH 8 or 7, it will remain the same. If there is no imine formation between P and E4, they will not hydrolyze also, and they will remain the same, and it will be completely transparent, no structures will form. However, if I have P and E1, they will form this PE1, they will form this structure. You have seen this earlier. Now, once they start forming these side products or byproducts or waste product, right? Side products or byproducts or waste products are forming. Now, these two are formed. This acidity of the medium will start cleaving it, start deprotecting it, because this is very acid labile. It will form this E1 and lead to more and more population. This is what you call classically self-sustenance, where the wear and tear of the molecule or the building blocks can be replenished by a suitable molecule that is in the system. In this case, the reaction is leading to deprotection of E4 or the acetal, and that leads to the aldehyde formation. And what we found out was interesting. We found out that when we added E4, E4 addition leads to more population of this, and the lifetime grew substantially longer. It suggests that it can self-synthesize also. The conditions can self-synthesize the building block, which is a hallmark of living systems, and that is what we call autopoiesis or autopoietic system, a very provocative term. Even the reviewers insisted on adding the term at the end. They were so happy with the concept of the word, where the molecules, these small tiny molecules, and I'll remind all of the audience, all of these molecules so far are less than 800 molecular weight or 900 molecular weight, not anywhere close to the protein structures or very complex proteins that are used, right? But these are doing it, they are forming the structures and they're breaking apart. But if there is a suitable precursor of the molecule, the reaction can lead to uncleaving or rather cleaving of this or deprotection of this, and that will unmasking of this in a way, you can say, and leading to this, and that leads to substantially, we can see from NMR also, substantially more time they are sustaining, and the population is also is more than the only the E1. So that was very encouraging to see that how it can self-sustain itself. Now the final piece, maybe or maybe last 15, I don't know, 15 20 minutes I have, or I have more time. Okay, so I'll carry on anyway. So there's a question in between. Shall I take the question now? Shall I take the question now, or shall we wait for the end? Okay, so Deblina Sarkar, Dr. Sarkar is asking, sir, in one slide we had seen self-assembled amino structure and equilibrium is actually shifting towards the self side where randomness decreases. So is there a temperature or heat factor? Well, temperature we have been keeping in room temperature, obviously all of them. But if I do heat it up, it's going to be very interesting to question actually. Heating will not lead to more self-assembly, right? Because self-assembly will be the soft assemblies have a melting point of around 70 degrees centigrade. So if I heat them up, in principle, it will not go on the right side, and it will be instead the imine formation will not be promoted by positive feedback. Maybe I'll detail it out in the last part of the talk, or when we take question and answers. But I will carry on now. Okay. Yes. So
Thank you, sir. Thank you.
yeah. So now this is the one more part of the story that is called liquid-liquid phase separation. I was talking about that liquid-liquid phase separation, or that concept of co-sorbate is very interesting concept here, because in living systems, even in advanced creatures like human beings, have this many organelles which don't have a membrane, from nucleus to this U body, P body, stress granules. Many structures are there which don't have a membrane, which is quite shocking. They are like liquid in liquid. So they are demixed from the liquid. You can imagine a liquid, let's say as water has only one phase, right? But if I imagine an experiment that water demixes and forms multiple phases, what are the phases? They form these droplets. Now, there's a dense phase and the dilute phase, but they are not like vesicles or a liposome, which has a charged structure. They are not colloidally stable, but in this case, these granules are these condensates are rather colloidally stable. I'll show you in just a bit. So this is really a hot topic, from PCs to biologists to origin of life concepts to medicinal chemists, or rather actual drug design people are also worried about how to mitigate and control any problems of the stress granule. But now, chemists have also talked about it, how to come and how to make liquid-liquid phase separation. Many good papers have come out, but all of them face one problem. And before I go there, the structures maintain their size. A nucleus doesn't grow and become a monster, right? Usually, you can imagine a liquid-liquid phase separated to be coming together and forming one phase or enlarged, but they don't grow beyond a size. There's a spatial stability. Okay, why is happening? Because they are doing a reaction coupling with that, and it's very interesting to see how biology controls and stops these two processes. What is the process? If I have a liquid in a liquid, they will become bigger by post-fall ripening. The smaller will become smaller, the bigger will become bigger, and they will become a very large droplet. At the same time, even if they don't show also ripening, they can really undergo coalescence. Again, coalescence is something that, as the term says, if you might have imagined kerosene drop in water, it will just coalesce to form this one phase, right? In this case, only one phase is there, water. The structures are forming and they are becoming these giant droplets. They become like monster, and then they fall out of the system, right? A very thin film is formed. So chemists have been able to mimic liquid-liquid phase separation. Professor Shomrat Mukhopadhyay and Professor Shomir Maji, IIT Bombay, Shomrat Mukhopadhyay, Niger Mohali are doing very good work on this field of intrinsically disordered proteins. So we work on small molecules. We wanted to see whether this can be done, right? Whether we can stop the growth of these liquid-liquid phase separation structures as a function of time. So what we did, we used this molecule. This paper was published this year. This molecule was used in aldehyde, again a very simple chemistry, and we added this peptide. We searched a lot, and we found out that this peptide in aldehyde, when mixed together, forms an imine, right? And it forms co-sorbates. Co-sorbates were growing. They're forming these droplets, very homogeneous droplets, not homogeneous, rather, sometimes they can be a bit more deformed, like typical liquid will be, right? They're coming together, sometimes forming a larger structure, and they were coming coalescence behavior to droplets. This is only reserved to liquid-liquid phase separated behavior, right? A vesicle will not do coalescence that easily. Those are energy intensive process, because a vesicle, as you may know, they are colloidally stable, right? Vesicles are or liposomes or micelles, let's say. A tiny micelle will not just mix together or will not come like two blobs coming to be a bigger blob, because they are charge repelled. The very existence of a micelle or or a vesicle or fiber per se is because of these structures are repelled to each other. In this case, they are liquid like, they can come together and form this, and they are can be also burned. Let's say you burn literally a hole on them by using a very high intensity laser. It's called FRAP experiment. They recover very fast. We suggest that in literally seconds or minutes, 2 minutes, less than 2 minutes, they recover to almost 80% intensity. We suggest that they are very liquid-like structures. These are two hallmarks of liquid-liquid phase separation. If you want to make something on the on your lab, in your experiments, it should not have more than any other. Only one phase should be there. It should not have any other DMS or something. One molecule can be added, one protein can be added. That will lead to this phase separated structure, which can be centrifuged to make a dense phase and a dilute phase. So when we did the control, if you have noticed, the sequence is a bit unique. It has AIB. AIBs are not known for their packing capabilities because they are a bit kinky, right? They have a sp3 carbon, although chiral centers do have that, but they have two carbon, two methyls like this, which don't allow them to pack very well despite the FF. So what we did, we added only alanine, and we found that rather than co-acerbate, they were forming fibers, amylase-like fibers, which was interesting to show that a structure is very important to have a something like a beta sheet breaker. We did now the fun part, how to control, because here we are seeing growth of the droplets. They were getting very big, very gigantic, and they were undergoing either oswald ripening or coalescence behavior to grow out of proportion. Now we wanted to couple a reaction, because as I said in biology, reaction is coupled to control the liquid-liquid phase separation. And again, I will repeat for the n number of time. It's a very unique and new field, and from physics, chemistry, physical chemistry, all backgrounds should be interested. Organic chemistry should be interested in this. If in case you are interested in supramolecular chemistry, this will really appeal to you. I'll request you to read more on liquid-liquid phase separation. But in biology, they control their size. The size don't grow and blow up out of proportion, right? As I said, instead, granule remain like a spatially stabilized system. So what we did, we added a reaction, again a textbook reaction, or the favorite reaction of students, aldol reaction. So what we did, we used a cyclohexanone. And the idea is that cyclohexanone and this can form an aldol adduct. Now, when the moment they form aldol adduct, they will not be fit to form the imine anymore. The imine was shown here, right? Because this aldehyde will be consumed with the cyclohexanone to create an aldol adduct. Indeed, when we added this, we found that the structures are now going down. This is one of the first finding in this, where we have seen coacervates now are not growing, instead they are shrinking inside and dissolving away. So that is not solving the problem in biology. In biology, we have co-acerbate or phase separated structures, condensates. They don't grow, they don't shrink also, they remain the constant size, right? So that was very interesting. At least the first step was done when we have coupled a reaction with a co-acerbating molecule, and we see that the size is going as a function of time. We did a lot of data. Obviously, this active system is this one, this pink one, which has a cyclohexanone. The passive system means there is no cyclohexanone, only the aldehyde and the peptide is there. We could see from HPLC, we did intensive or extensive HPLC, and we found out that indeed there was a lot of aldol product generation. We found out interestingly that when the co-acervate phase, that is a P1, that is with the AIB structure, the co-acervate phase, the rates were higher. However, when the fibers were used, which is this P2 alanine, the rates were significantly lower. So the co-acervate phase, for some reason, leading to more and more activity than the P2 phase. Okay. Here was the fun part. We started now refueling basically, or rather, excuse me, keep adding on and on. Okay, so what is happening essentially is that, I will repeat again, P1 and ARCHO can form this imine, which can form co-acerbate. So far so good, but they can grow, enlarge, become monsters and settle down. Expected. All papers have shown that in chemistry journals. However, if I add a cyclohexanone, the gray dot, it shows a reaction, and hence the P1 is free at the end, and the aldol adduct doesn't participate any more imine, and it doesn't form any coservate. Indeed, when we added one more batch of this blue, pink, and gray dot, basically the aldehyde and ketone, we found coservate to be forming again, but then they are again vanished. Again they are dissolved basically. Again, we can do that multiple times. This can be done. Interestingly, the size was maintained also. And if I do a passive coservate, by passive, again, I'll repeat, we mean we don't have the cyclohexanone, so the reaction is not going on. Only reaction that is going on is the reversible imine bond. So when we added the aldehyde and P1, we kept on adding the aldehyde, the pink dot or whatever this color is, this color we keep adding. You see that they are getting very big, and confocal cannot catch it because they are falling out. They are becoming very big, and they are forming this film in the confocal microscopy slides, and we don't see any image. Interestingly, now we did just like biology and open system. Biology is open system, right? We have constant exchange of molecules in the cells so that the structures are maintained. So we asked that if you have an open system where we have a passive system and an active system, again, the passive system will be only the aldehyde and P1 peptide, active will be aldehyde, P1, and the ketone or the cyclohexanone. And we found out that, and you can see from the video, the passive is settling down very fast. Although they look to be transparent, they're settling down very fast. However, at the same time this is also running, they have a substantially longer lifetime. Remarkably, we found out that their lifetime is not only higher, their size is also maintained. And this is a classical example of non-equilibrium steady state. What do you mean by non-steady state? If we keep adding the fuel at the right point, you will see the sustenance of the structure, right? Just like I don't know how many of you are organic chemists. You see a reflux condenser, right? You have inlet, outlet. You maintain it so well that you look like if the condenser is empty enough, you'll see nothing is there inside. Everything is in static water condition. Actually, it's flowing, right? The flow of inside and outside is same, so it look like to be exactly in a static or a stable state. It's like a Casey-Nag mathematical problem, right? We used to have this tank and a whole concept, right? The water entry, water exit. Here, the fuel is being added. The fuel, in a way, you can say the reaction is driving this. The ketone and the aldehyde forming the aldol adduct. The aldol adduct is leading to no coservate, but we keep adding the two, and then we keep maintaining the size as a function of time. Okay, so this can be shown in this cartoon. Only this aldehyde will lead to coalescence, but if I keep adding this aldehyde and ketone, we see this happening, and if we add them in a right sweet spot, kind of a addition, we see the formation to be maintained as a function of time. The sizes are maintained as a function of time. All right. So in the last maybe 5-10 minutes, I'll talk about maybe 5 minutes. I will not talk to for too long. We will take questions more.
Yes, sir, you have time.
We have time, right? Okay, good. So this is the last part of the story, and here we talk about this movie. This movie is about 1966. Many of you and us were not born, right, that time. But the movie is very unique and really touches the imagination of any individual. The movie says that you have these micro structures, the micro robots, in a way, which is basically rockets and these spaceships, which will be very, very tiny in their blood vessels, and they will have these tiny surgeons, complete science fiction, right? Science fantasy. And this surgeon will go to a particular site, let's say a liver or a urinary bladder or a brain or a heart, and do the surgery, and they'll come back. It's called Fantastic Voyage. This movie has been coming up back in up in science because of a field called micro robots. It's a really hot topic again, and it's also direct derivative in a way of systems chemistry. But not, it's non-equilibrium. That's why. So what is that? So that's what the cartoon shows. Let's say when we are in a home, we know where the kitchen is from the smell of the food, right? We can smell biryani from pretty far distance, especially people from this part of India, that where the biryani is being cooked. That is called chemotaxis. Why chemotaxis? Chemo means chemical, taxis means moving around. Just like aircraft does this taxiing on the runway before taking off. We do a chemotaxis. A biryani smell can lead to us to the kitchen. And similarly, let's say a dustbin is there, a very, very miskept dustbin. You'll try to go away from that. The first part will be positive chemotaxis. You're going towards the chemical gradient. The next part will be negative chemotaxis, where the bacteria is moving around, moving away because it's not liking it. It can be also phototaxic. You can turn on light, you see any lunch is going on, there's too much of sun, everybody will try to go under the shade. Or the bikes will be parked below the trees, not outside, because that is negative phototaxis, right? It can be for positive phototaxis also in a winter, a very cold winter, you will try to be in the sun. So I mean, it's a systems level concept I'm talking about, but it's very, very profound, right? And here also I showed the video earlier how the bacteria was trying to hound this supercritical. So we were really interested in that whether we can create, let's say, a nano rocket or a structure, let's say a vesicle or a nanotube or peptide nanotube or a safe biology compatible structure which can really sense a chemical inside a body and go there and do the job, right? It's called micro robot in a way, microstructures acting like robots. So we were really, enthused with this idea, and we saw that, yeah, these are one more video where they show movement, motility. There also you can see this is the probably the food source where the bacteria are showing motion. So yeah, even a very swarming behavior is seen in biology, where you see the school of fish all going together in a school, right? In a swarming behavior. Also, sometimes birds do that. Very large amount of birds are together. Large thousands of birds are going in one side, right? It's called collective behavior. It's very, very unique. So this is the one of the first paper that came out. Actually, Aishman Sen, this part from Indian Bengal origin, in Penn State has a lot of contribution on micro robots. But usually his systems are all nanomaterial system or metal nanoparticles based system. But we wanted to use biologically compatible system. So what we need a rocket, we need a chassis, just like a chassis number is there in your car, you might know, in your bike also the chassis number should be there. So a chassis you need an engine, right? Engine will be the car engine or the bike engine, and you need fuel, right? A fuel that will be used for running the car, or the fuel can be sensed by the car to go in. This doesn't make sense in that directly, but just like, let's say, even the simplest of life forms can show cognition behavior and go to a food source, right? So we need all this together, and what we did, we used nanotubes. These were published earlier by many US paper from our group. These are all published as catalysis or various journals in terms of catalytic activity or chemistry journals. But we use those structures again, and we added enzyme on top of them. The idea was that if the enzyme is there, I did not go in detail on that because this will be a completely different story and took 1 hour. So when we added an enzyme, let's say UDS, and we have other enzyme called cytochrome C, which can sense pyrogalol. So if you have a gradient, it goes to that direction. So the nanotubes will smell and sense the gradient and will start growing there. So we could see that as a function of time. This paper also showed that not only enzymes like UDAs, also catalase, they are, sorry for this, it's not showing in all the structures, but catalase enzymes converts H2O2 to O2. You can see that from H2O2 to O2, this will lead to bubble propulsion. Just like a bubble coming out of a object will lead to a motion, these structures were seen to be moving. So I'll show you the structures here, and you'll see that how they are moving very fast. I mean, we can tag them and we can see them. This is also moving. I did not show the control. I should have shown the control. The control shows only some Brownian motion, something like, if you can see my video feed, they're just oscillating or moving around that. In this case, they're moving substantially to long direction. Okay, in this video also, if I do, let's say, in this video also, if I do a control without the chemical fuel, we see very less movement, but here the movement is very, very high. The idea is that what can be useful, I'm in a talk. Yes. So what can be useful is that this can be really used to locate a particular site and deliver that idea. So we are doing now these microfluidic studies. We can see that in case if we have, let's say, two gels, one gel having the chemical, it can be H2O2, it can be pyrogalol, it can be glucose, it can be urea, which is very important. Glucose, urea, H2O2 are all used in biochemistry. In fact, urea can be used for urinary bladder targeting. Glucose can be overexpressed in cancerous cell. H2O2 is also overexpressed in cancerous cell. So can be, we have seen there's a telltale sign of movement of left to right. Only on this direction the motors go. Just like we smell food, go to the kitchen, the structures smell and sense these chemicals and go towards a particular direction. So what can be this be used for? This is just snippet from a proposal that we submitted earlier and got funded. It is basically having, let's say, we can add enzyme, ureas, and we can add radionuclide therapy. That is this radioisotope level sodium iodide is known therapy. But if you mix them together on the surface, it can actually be sensed towards where the uric acid is generated, and then this NAI will lead this isotope level, or rather, this radioactive NAI will lead to toxicity of the cells, and the cells will die. And similarly, similarly, this can happen for other subjects also. The point I'm making is that it can actually lead to this old diagram, very old movie of let me show you this. This one where we have this nanotube as the structure, where the microsurgeons will be removed and directly the medicine will be added, either a drug can be added, and the motors, the structures will move towards a particular gradient. So what is the advantage? Usually nano medicine has a typical problem. When it is injected in the bloodstream, they can undergo sedimentation, because the capping agents can be ripped off and fall off, right? They will start falling to the ground, or rather, in the context of biology, it will be sedimented in the particular organ that ideally you don't like it to sediment for, right? So then here in this case, these are active swarming structures which can indeed be always moving around, and that will lead to more penetration and will sense a chemical and go to the site and deliver the object. So these are essentially active material which otherwise you cannot be, you will not be able to access this structure, this activity in the absence of active material. Okay. So finally, I'll also show that what is the advantage of the other structure that I talked about earlier, the transient self-assembly. I'll show you here. It's a very busy slide. What I showed you earlier, sol gel, sol gel, sol gel. So now we are talking to various companies, and Origin Oncology was very excited to see this kind of gel sol behavior. It's like a hydrogel you use, right? Or a hair gel we use to set our hairs, or maybe a face cream, which are usually gel. But in this case, the gel is breathing, right? It's forming sol gel, sol gel, sol gel autonomously. We don't have to do any external tinkering with that. So what we are planning with this company is that we have a dermal patch which will have this gel. Now this gel will have a drug. Now this drug will release very much, will be released in the bloodstream when it is in sol state. However, in the gel state, you will stop releasing it, or the release will be sustained. It will release very fast and then sustain, release very fast and sustain. That part will be sort of an on-demand delivery vehicle, or delivery platform, which is quite remarkable, because this is only possible, this kind of really manipulation in terms of demand specific, or as a function of time, changes in terms of the drug delivery or the release of the cargo, is only possible when you break the constraints of equilibrium, right? So here also we can see that in principle, these functions can be unraveled, which cannot be imagined. Even the micromotor or nanomotor project or nanorobot project we showed you earlier, is funded from DBT. That is also possible when you break the constraints of equilibrium. Okay, because here as a function of time, you can see the change of the drug releasing and holding capabilities, which is the function of time term. The term called function of time is usually not used, not related to equilibrium. Equilibrium doesn't have a function of time. Usually it will be stable for a long time, right? But in this case, non-linear structures will be active systems. It will be stable as a function. It will be showing or a difference of function or a tunability as a function of time rather than autonomously. From the micro robots, one example I give you, the other example is this one. Similarly, we have seen that if we imbibe an enzyme, we see the enzyme activity to be really oscillating. Very high activity enzyme because of gel networks, and then low activity. Then high activity, low activity is happening as we speak. The sol gel changes are going on. Okay, so with this, I slowly wrap up. The systems chemistry is a very new topic. This is not only exciting to chemists, but it is exciting to physicists also, biologists, and chemical engineer, because one of the experiment I showed you that open systems are typically done by chemical engineers. Now the lines are blurring anyway. So if you are interested, you can read these reviews. Systems chemistry is a very emerging field. It started in Europe. First Gordon Research Conference happened in 2018. That means that tells you it's not even 10 years now. That tells you how new and nascent the field is. Because Gordon Research Conference usually, for maybe chirality or maybe some organic synthesis, it will be for maybe last 60, 50 years. It's only 8 years old. That shows the emerging field of systems chemistry. And we do keep doing conferences in terms of to expose people to the concept of systems chemistry. We do this online event, particularly during the COVID time. We did every once in 2 years. Nobel laureates spoke about it. This is an online event done with Azad Kolkata and other institutes like UCLA and University of Strasbourg and University of Ulm in Germany and Strasbourg in France. And also, as the person who introduced me talked about, that I will be organizing Gordon conference in the next year with Elisa Franco as a co-chair and as a chair in 2028. For your students out there, if the students are interested, go for the Gordon conference. There's a lot of opportunity in terms of funding supports, especially for girl students. They can apply for funding. Their registration can be completely waived off. All they have to arrange from a CRB or NRF the travel. So that can be there. No guarantee though, but usually in the bigger, larger Gordon conferences, that has happened. Many of my students apply for the grant, go there, and they say that they don't need the money. One PMR student also said that because they got funding from the Gordon. So that kind of opportunity is also there for all Gordons, not only this. This will be in the USA. And with this, I close also in terms of by thanking the students. They are the pillars. As you know, any lab will only succeed if they have passionate, hardworking students. And the funding agency is primarily Swarnayanti has been a very good pillar last 5 years. And Nano Mission, all those grants, MOE Stars and Azad Kolkata has been very kind. Special thanks to Shobik Maiti from IGIB and Amita Das from Azad Kolkata, two rather senior people who have been support to the lab. And these are students who have really contributed. And thank you for your attention.
Thank you, sir. Now this session is open for discussion. If you have any question, you unmute yourself and ask. Any question? Okay, sir, I think there is no question. So thank you, sir, for very informative and insightful presentation.
Thank you.
You have taken us to the heart of system chemistry, showing how life essential function, replication, metabolism, and compartmentalization can be synthesized and integrated under far from equilibrium conditions. Your insights into the reaction network, self-reproducing systems, and transient compartment have clarified how chemically constructed systems might begin to exhibit lifelike evolving behaviors. We are grateful for the clarity and vision with which you have explored this fundamental question about life's emergence. Your work inspires us to continue pushing the boundaries of how simple chemistry can be guided towards complex, dynamic organization. On behalf of everyone here, thank you once again for your prestigious Bhatnagar Award in this year and for throughout thought-provoking and forward-looking talk. Thank you, sir.
Thank you so much. Thank you for having me. Thank you all for the wishes. Bye-bye.
Thank you, sir.
Yeah.
So with this session is in for today. You can leave everyone.